The overall goal of this procedure is to prepare an activatable liposome that enables in vivo imaging of inflammation with a high sensitivity. This is accomplished by first preparing a lipid film composed of phospholipids. In the second step, spontaneous vesicles are formed by hydrating the phospholipid film with a quenched concentration solution of a near-infrared fluorescent dye. The spontaneously formed vesicles, or SFV, are extruded to ensure homogeneity of the liposomal vesicles, and then the liposomes are purified from the residual non-encapsulated dye molecules. Ultimately, spectrometric measurements of absorbance and fluorescence emission are used to show the quenched state of the encapsulated dye, while in vivo near-infrared fluorescence imaging is used to demonstrate the potential of the liposomes for imaging inflammatory processes.
The main advantage of this technology over other existing ones is that we can determine the signal directly after internalization of the contrast agent into the cells. That means that they are activated in the cells so that the signal what is detected comes exclusively from the cells.
The implication of this technique extends toward diagnosis and therapy of cancer, since phagocytes such as tumor-associated macrophages can take up and degrade the liposomes, releasing therapeutic drugs or imaging probes within the tumors.
Generally, individuals new to this method will struggle because the encapsulation efficiency is influenced by the properties of the dye used and the effectiveness.